So thank you very much. Hello, everyone, and really appreciate this chance and for Quanlin's invitation to talk about my my research. And today, my topic is to leverage multi-omics data for disease genome mining. And and thank you for Quanlin's introduction. I graduated from USC and had been doing my postdoc at Yale. And the last it's already last year in July I started my own lab at the University of University of California at Irvine. And I have been working in ENCODE for many years in the data analysis center. So for this talk is heavily focused on ENCODE data processing and how we're gonna use the annotations there for disease genome mining. So let's go to it. And so my major interest is in the human genome, and I want to check what are the implications of different variations across the population indicate. And we know that they sometimes, most of the time, they indicate some variations from the phenotype level, but we do notice that they are responsible for a lot of like disease status. And when we look at the huge 3.2 billion base pair human genome, we know that not every nucleotide is equally important. And some part we know that we know that better. They are the coding regions. They are more than less than 2% of the genome, and we can see their products directly. So we have a pretty good interpretation of the functions of those regions. But the rest of the genome was considered as the dark matter of the genome, but they host over 90% of the disease risk loci, and they contain extensive regulatory information. So my research is to investigate what are the possible functions of different non-coding regions in the genome. By doing their functions, we were trying to integrate some genomics data to inform which regions has been bounded by transcription factors, which region has been transcribed, which part of the chromatin has been open, and what is the 3D structure of the chromatin. And by inferring these functions, we are trying to integrate quite a lot of sequencing data. So my work is to process the sequence data and then give us a map to the function of the non-coding regions. And then when we are trying to link the nucleotide level variations to disease status, actually we are having a very challenging task. One of the major reasons is we are having 3 billion base pairs, so they are pretty huge, and we were looking for one nucleotide or a couple of different combinations of genetic variations to be responsible for the phenotypic variations. And this is actually looking like we are having a lot of candidates like in this problem. My son's favorite game was to look at the panda inside the snowman, but we have 3.2 billion base pair pandas, and we are looking for the snowmans. We are looking for the only one panda that has been responsible for the disease status. So when I was looking at this and we look back of the previous annotation efforts, what we did is we're trying to uniformly annotate the genome. We are trying to go aggressively to know each nucleotide, what is their function, and we are trying to be as comprehensive as possible. But on the contrary, when we are trying to use these annotations to do disease genome mining, actually we want to be as concise as possible, right? We want to build a very, very sharp annotations that we know that they're high level, high accuracy, like annotations, and they are pretty sharp. We know where are the real function site to better map the disease causing variants. So considering this less is more impact in the genome annotation for disease genome mining, we actually have tried to build an annotation resource within phase 3 of ENCODE that is called ENCODE. For this ENCODE annotation resource, actually, we are focusing on some like tier 1 cell line data that has been sequenced very, very deeply, and that has been comprehensively sequenced. We have quite a lot of functional genomic assays. We want to build the non-coding regulatory elements, but on top of that, we want to link them to the genes. So we have the distal regulatory elements and their accurate linkage to genes by integrating quite a lot of epigenetic data and high-C data and some other source of information. For instance, with EQTLs, we use them as the validation to evaluate our enhancer to gene linkage. And on top of that, we are trying to construct different networks, like the networks for RNA binding proteins and transcription factors. We organize these heterogeneous networks into different network hierarchies so that we can measure the network dynamics, the network changes across different stages. We're going to introduce some of our computational work there. So the center thesis of the talk today is about how we're gonna contribute, construct such a condensed, compact genome annotation, and how we're gonna apply it to prioritize some risk factors for disease, especially cancer, in today's work. And let's go to the first part. So first of all, I'm going to talk about some of our computational pipelines to define compact regulatory elements. Specifically, I'm going to talk about enhancers in this work. So previous enhancer predictions, there are many, very famous algorithms, for instance, QuANT-HM and other methods. But that was back to around 10 years ago when we do not have quite a lot of training data. We are mainly confined as the unsupervised training algorithms for this enhancer discovery problems. So at that time, the resolution of our annotation is completely defined by the input. If you give me an input of 200 nucleotides, then my resolution is 200 nucleotides. Or if you give me very, very huge regions, my annotation will be pretty huge. And that was actually a binary classifier problem. So we have been working in the ENCODE, like a functional validation center, and where we can actually access quite a lot of whole genome, parallel reporter assays, especially star-seq data. So our thinking is, since we have quite a lot of star-seq data, can we use those data to train a deep neural network to find some very specific patterns? For instance, if you look at the histone modification marks around these star-seq peaks, you immediately see, rather than absence or existence of some potential epigenetic marks, we can see there is a obvious shape, like a pattern of the true enhancer regions. You can see in the middle of the enhancer, the chromatin is open, and then there are some transcription factor binds there. Because of the binding events of some transcription factors, when we were doing the ChIP-seq data, we usually can see a dimple through these two peak regions that we call it the double peak regions. So these specific patterns can help us accurately identify enhancers. And so what we did is we collected the whole genome star-seq data from different cell lines, and then we also collected matching histone modification marks, and also the chromatin status data. So instead of a binary classifier as most of the previous methods, we are training it like a object detection problem. So you can give me any input region. It can be 1 kb, 5 kb, it can be of any length. So what I'm gonna do is I'm gonna answer two questions. The first question is whether there is an enhancer inside this region. And the second problem is I can have a score to indicate which region is important for me to make this positive decision. So in that sense, I can have a per nucleotide resolution classification about whether there's an enhancer and where is this enhancer. So to do this, we actually decode one method that we actually develop one method that is called decode. We use like star-seq data as our training data, and we extracted the epigenetic profiles from ENCODE, and we input it into a ResNet, deep convolution neural nets. And then we use GradCam to assign a score at each possible nucleotide, how important it is for us to make enhancer predictions. So as a result, for all the positive predictions, for the ones that we think that contains an enhancer, we have a core region, core enhancer region definition. So in general, we shrink the input size to around like 13 to like 15% of the distal regulatory elements. In this way, our enhancer definition is pretty sharp, is usually 100 to 130 nucleotides. And this is very important when we are trying to map the genetic variance into these functional genomics regions. And then we validated our enhancer definitions using the mouse transgenic assays. That's one of the data set that has been used in the ENCODE enhancer challenge. And we benchmarked our method. Oops. Our method is called like decode, that is this orange bar here, was the previous leading method in the ENCODE enhancer challenge that is called match the filter. Actually, we can outperform the existing method in almost all tissues. And we also compared like how important it is to condense these enhancer regions. For instance, for the distal regulatory elements, we shrink it to around 18% of the different input regions. And from the proximal elements, the shrinkage size is a little bit different. So we have around 68.6% of the original input size. As you can see, the condensed regions as compared to the original positive inputs, they are conserved across the populations and they are conserved across species, which indicated that actually they are of higher quality as compared to the raw inputs. And then we also run LDSC to check the GWAS variant enrichment in very consistent phenotypes. For instance, in this series, we train our model in ENCODE cell line data, and then we predict on neuron progenitor cells. And we checked the enrichment of the condensed region of the GWAS variants in the condensed region versus the original region. We'll see using only around 18.7% of the genome regions, we actually achieved higher GWAS in variance enrichment. So that's our first work. So by introducing this object detection problem in a canonical enhancer prediction problem, we are actually able to confirm very, very compact genome annotations that hopefully can increase our statistical power to discover the causal variants for some disease. That is part one. And let's go to part two, that is disease genome mining. I see there are some questions in the chat probably. And so Quan-Lin, are we gonna handle these questions during my talk, or we have a separate like a session to answer some of the questions along the way? What is the best way to handle the questions?
Jing, it's your preference. I think it might be easier to handle it all in the end, given that you have, a relatively short presentation.
Okay, sure. Yeah, okay. So feel free to ask questions and I will answer them at the end of the talk. Yeah. And the second part is how we are gonna prioritize. So we have collected many functional genomics data, and then how we're gonna prioritize like the risk related regions. And the first part is to prioritize the regulatory elements. So one of the application is how we can use the ENCODE data to help us to find the potential driver regions of cancers. So this application, we use the somatic mutation test for cancer like associated driver detection. So for this test is actually we pour many variants together and then we have a test region potentially that is our functional annotations. It can be enhancers, promoters, or it can be the enhancers plus promoters plus the coding regions as a whole as a test unit. And then we are trying to have a decision to see, we compare the background, that's our expectation, and we compare with our observed data. We are trying to see whether there are more than expected mutations in those regions. And but this is a challenging task, mainly because we see quite a lot of heterogeneity. So for the first version of this method, people are using a constant mutation rate assumption, but we know that this constant mutation rate is violating our observations. There are heterogeneous mutation rate across different tumor types, different samples within the same tumor, and different regions of the entire genome. And then we are trying to collect a wider range of data and fully prepare those data to help us for a better background mutation rate prediction. So what we did is we have collected the entire ENCODE data, and then we use those ENCODE data to predict the background mutation rates. And because we found a lot of epigenetic features has been correlated with the background mutation rate. So as you can see, the accuracy or the correlation between the observed and the predicted a background mutation rate has been progressively increased by the more genetic features that we input in this model, and we can see the importance of different epigenetics marks to how they are affecting the mutation rate, how they are affecting the important the prediction model. So over these years, we have developed different models. So as compared by the constant mutation rate, we actually first introduced a variation like a parameter into like the P, which is the point mutation rate for each nucleotide in the genome. And then later on, we are using a regression model to predict the mean of the P value here. And in this way, this V1, V2, Vk, that are the K factors that we can extract into from the ENCODE data. We pre-process all the data, we organize it in a data table. So users can directly download the data table and we provide a model to predict the background mutation rate by incorporating those functional genomics data. And then for our models, you can see if we have a p-value of different annotation regions in the genome, we actually significant by introducing this background mutation rate correction, we are like improving the inflation of the p-values that is due to the inaccurate mutation rate modeling. And in this way, we are reducing the number of false positives when we are discovering the potential overly mutated regions in our annotated genome. So that is the first part. By integrating the ENCODE data, we are trying to find the regions in the human genome that are truly under like a higher somatic mutation burden. And then I'm gonna talk about our two thinkings about how to use our annotation to prioritize the regulators. Here by regulators, so I mean like transcription factors or RNA binding proteins. So the first part is we have constructed this network from transcription factors to target genes in both tumor cells and normal cells. And then we want to ask a question that what has been changed. So in this specific example, let's see on the very top are the regulators, they are transcription factors. On the very bottom, the squares, A, B, C, Ds, they are basically the target genes. So we calculate the difference of this like tumor network versus the normal network. And then we immediately see on transcription factor 2, we probably want to follow up because they have gained quite a lot of edges. But in the blue like example, that is transcription factor 3, we also want to follow up on it because they lost a lot of edges. And we are not quite like interested in transcription factor 1 from this aspect because there's nothing has been changed. So instead of a naive like a counting algorithm, we actually used some community or topic detection techniques to measure these like transcription factor changes, network changes between two conditions. Our intuition is the original network between the transcription factors to the target genes. They are very sparse, they are very, very noisy, and they consider each of the target genes as independently and equally contribute to our revarying index. But however, we know that these target genes are not independent of each other. They maybe belong to the same pathway. So even if there is some jump, it's not as like a high impact as the jump is happening across different pathways. So what we did is we want to introduce the gene topics at the middle layer. And the gene topics was actually able to capturing this pathway information. So we are not marrying the direct change between the target, the transcription factors versus the target genes. Instead, we are marrying the change between the top, on the very top, the transcription factors versus our gene topics. So what we did is we actually use the LDA model. So we put the TF regulatory networks of the transcript, the normal networks versus the tumor networks together. We have a giant matrix, and then we input each of them into the LDA model. So in this way, in this sense, we are considering the target genes of a transcription factor is actually a document. And then we were detecting gene communities or the gene topics to mimic the pathway information. We're going to do a dimension reduction to transform the original huge matrix that is around 50K genes in total into a topic by different transcription factor like matrix. And then we look at one transcription factor. For instance, for this simplified example, we know like for this orange one, this transcription factor, they jumped quite a lot across different genes. And we want to follow up with this one because they have a high revarying index. And for this like a green one, because we think their topic distribution for this transcription factor is very similar. So we tend to not to follow up on these specific transcription factors. And then we applied this LDA technique on like K562 and GM12878 as a like a proof of concept application to investigate these network changes between two conditions. And in this condition, we are roughly matching it to tumor to normal to tumor transition. So in this one, in this application, we have collected 109 transcription factors. We prioritize those that has high like a rewiring index. And some of them, for instance, quite a lot of like tumor suppressor genes like IKZF1, PML, NBN, they were in the loser group. That means in the like a tumor state, they actually have lost a lot of activities in the regulatory network. On the very top, like for the Giner group, MEEK and NRF1 are the two like most significant winners from the K to from G to K transition, and MEEK, and we found a lot of together with other like oncogenes, they are falling into this group. And then we also think about like introduce the transcriptome changes and combined with our network information to try to find the transcription factors and RNA binding proteins that can drive the very specific shape of expression profiles in the tumor cells. So what we did is actually fairly simple. Our thinking is we have the transcription factor like a regulatory network. It can be a TF regulatory network or it can be a RBP regulatory network, depending on what network you choose. So if there's a check, it means it is connected to the target genes. So what we did is we just sort this target gene into like the upregulated gene versus the downregulated gene. And then in this case, in transcription factor 1, we do see a very unique pattern. Its target genes are consistently upregulated. We think, okay, probably we want to follow up with this transcription factor. Or on the other hand, we see transcription factor 3, their target genes are enriched in a group that has been downregulated. We'll see, okay, TF3 may be playing an important role. And for TF2, because we can see a very clear pattern, so we think we're not going to follow up in TF2 in this like a scenario. And then we did this for all the patients, and we get a summary statistic from each patient. And then we see in how many of these patients there is a clear pattern for each of the transcription factor and RNA binding proteins. So what we did is we collected over 2,000 ChIP-seq data and 459 ECLIP data. And then we ran a model selection using different transcription factors and RNA binding protein data and try to use them to predict the differential expression data from different cancer types from TCGA. And this is our discovery. And we prioritize the transcription factors in specific cancer patterns. And we immediately see actually in MYC, its target genes is mainly consistently upregulated in most of the cancer types. But on the other hand, like we use the MYC regulatory like activity score in the patients to see how dysregulated the target genes of the in terms of differential expression in tumor and normal cells. We can see the mixed regulatory score can separate the patients from breast cancer into distinct groups that they have different survival levels in our analysis. And we also look at the RNA binding proteins. So we find that sub1 is actually an RNA binding protein that has been enriched in the 3' UTRs. And then they actually control the stability of their target genes by binding to the 3' site. And we also find it has a pan-cancer, like a consistent pattern. Most of its target genes has been upregulated. And similar to MYC, this is in lung cancer. Actually, we found we can use the activity levels of sub1 to like separate the patients into different groups that has some like indications of the patient's survivals. And from these two methods, that's how we can select the key transcription factors that they are playing a larger role in cancer patients. And then we also zoom into the nucleotide levels, and we are trying to see if we have a very deep and compact genome annotation, can we find the nucleotides that has a larger impact in terms of the gene regulation. So one of our effort is we developed this tool that is called RADAR, that it can give you a functional impact score for any input variant in terms of dysregulation for the RNA binding proteins. So previously, we do not have large profile of the binding events for RBPs. So in phase 3 of ENCODE, there are, at that time when we did this work, there are 350 ECLIP experiments for 120 RNA binding proteins. And this number has been grown to like over 400, I think, recently for the ECLIP data. And for this data, we can see there are approximately 1.5 times the size of the exome. Is actually pretty huge regions. If we break them down into different functional regions, we'll see around half of them has never been annotated before, although they are mainly in the intron regions, the exon regions, and but their functions are largely unknown like previously. So, but we see that for each of these previous categories, as long as we can see there is an equally binding size, they are more conserved across the population. They are not allowing like a lot of genetic variation to happen in those regions. So that's a high and a very strong indicator of function for those regions. So what we did is basically we look at the fraction, we merge the genetic variation across a large cohort of population. And then we calculate whether for those RBP regions are enriched for rare variants. If they are enriched in rare variants, that's a strong indicating the functionality of this variants. So you can see this is the blue dot is our observation, and the yellow dot is our expectation of the percentage of rare variants. And we can see that almost for each of these transcription, these RNA binding proteins, we see they have more rare variants as compared than our expectations. So in other words, we believe they are highly functional. So what we did is we use like entropy weighted scheme to assign a score for each of the variants by considering different features. So it can be whether it's a binding events, whether this is a conserved structure regions, or it's in a like breaking a motif, or it's associating with key RNA binding proteins. So by this weighting scheme, we are trying to give an important scores for each of the nucleotides to evaluate and quantify its impact in terms of RNA, RBP dysfunctions in the genome. And then in the end, we actually combined our element level, our regulatory level, and our nucleotide level computational methods into a very coherent pipeline. We are trying to prioritize the key genetic variants that has been some related with a phenotype that has been related with a genetic function. So in this way, we proposed 8 different genetic variants, and we worked with Dr. Kevin White's lab at campus. And we use Lucifer's assay to validate those variations. And we found that for the majority, for the 6 out of our prioritizations, for this a single nucleotide genetic variants have profound impacts in the target genes expression levels. And this is one example. So we took a genetic loci, and then we can add any most of the genetic functional genomic region assays and our annotations to see what has been changing if there's a mutation that from C to T mutation, what's its impact into its target genes, and what is our possible explanations for this. And for this specific example, that is in enhancer region, that is an enhancer region that this mutation can introduce a motif disruption events that it can change its target genes. We also, in our ENCODE paper, we also have some other examples that are nucleotide change in the insulators can help block the connections between the enhancers and target genes that can introduce some phenotypic distances. And that's my published work. And I wanna recently introduce some of our current work in a single cell. And we are trying the same thing using the single cell multi-omics data to construct the regulatory grammars in a cell specific manner. We are trying to identify very, very compact annotations in the human genome, so we can actually look at the mutation impacts very accurately in a cell type specific manner. So our first work is how to integrate multiple samples of single cell ataxic like assay for very accurate cluster events. And then we use the variation autoencoder, but we introduce artifact that has been caused by either the relapse or which batch of experiment it comes from. So we actually introduce a batch effect correction and confounding effect correction into our architecture of variation autoencoders. And this work has been called Sailor, and we make it online, the preprint version online. And please, and we're going to release the package for this Sailor method for single cell ataxic data processing very soon. And also we have another method that is called link-seq, that is to link the enhancers to promoters very accurately. And then we combine the link-seq module with our previous, we call it DART at that time, DART module to define the compact genome annotations, so that we have a very compact extended gene annotation that includes both the coding regions and the non-coding regions. Our link-seq module can work on single cell ataxic alone, and it can also work on the single cell multi-ohm data. So that's our some recent progress in defining the annotations of the human genome, and we are trying to apply it into disease studies in a time-specific manner in the future. And I think that's roughly my talk. And I want to thank my advisors, Mark Gersten at Yale and Dr. Liang Chen at USC, and many of my collaborators and my colleagues to work on this project. Thank you. And any questions are welcome. Thank you.
Thank you, Jing. I think given we have a manageable audience, anyone, if you have question, feel free to just directly unmute yourself and speak up. If you can't, feel free to let me know.
Hi, I have a question.
Yep.
I'm curious as to your thoughts about, you talked about identifying like if there's an enhancer and where is that? And then you talked about creating networks from transcription factors.
Yeah.
What are your thoughts about like taking GWAS data, identifying those enhancers, and then moving to the network, kind of like going backwards? Is that something that's in the DECODE code?
Oh, yeah. I think that's a very good question. And we are doing that. The thing is, when we are having the GWAS variants, we are trying to find all the variants that are in the same LD block. And then we are overlaying the entire LD block with our enhancer regions to try to prioritize which regions actually has the high impact in terms of the final phenotype. And then we are also thinking about not to just search for the given vector, because for our nucleotide impact quantifications, we actually have a score for all possible mutations in the genome, what is likely to be most profound in those region. And then we work with the experts, for example, Dr. Kevin Weiss' lab, to validate our priority variants. It may be a common variant that we have seen in the same LD block, or it may be whatever possible mutation that is not even seen in the patient that's in there, to maximize our chance to really capture a function nucleotides in those regions. But the thing is, let's say if we have very huge enhancer, let's say if we use the raw ataxic peaks that can spanning thousands of nucleotides in length, there are some long peaks like that, or we have a lot of enhancers that has super long. When we are overlaying it with the LD blocks, we actually end up with a lot of enhancers. We did see this for a number of psychiatric disorders. For instance, for schizophrenia, they have a lot of GWAS loci. And when we were considering the whole LD block, we end up with like over 100 enhancers, but if we don't have a like a a high throughput assay, it's very difficult for us to decide which enhancer is more important than others. That's our original thinking, that we need to shrink the size of the enhancers and remove the ones that we are not very sure, or we are integrating multiple individuals. We call it, sometimes if we have the data, we calculate a cross population conservation score for this functional annotation. We only choose those that has been conserved across many people, that we think they are really highly functional, they are very reliable. We call it probabilistic annotations. And in that way, we are hoping that we can reduce the number of prioritized regions so that we can do focused follow-up in those regions.
Thanks.
Well, as we wait for other people's question, I want to ask some questions too.
Yeah.
So yeah, Jing, as you said, I think the epigenome really helped us funnel like the huge non-coding region, right?
Yes.
And I actually have a reverse question. What's the implication for the coding region? And when you presented on the somatic mutation rate correction,
Yeah.
I assume that most somatic mutations are detected in whole exome sequencing data, which is mostly in coding region, right? So in coding regions, how does epigenomic mark help you prioritize certain signals? What's the best uses there?
Well, that's a very good question. The thing is, when we are trying to define the mutation burden, even in the coding regions, in general, we would like to use a very local, like a background mutation rate. That is, if we use the entire genomes, like a background mutation rate is actually pretty heterogeneous. So what we did is we just chop the genome into different like blocks. And in each of the block, actually, we are trying to fit a model, like using a negative binomial regression, right? We use the input as epigenetic marks as the input X, and then Y is our background mutation rate in this local block, right? Even in this coding regions, I will say we have to consider this epigenetic marks. Of course, there are some other high impact. For instance, the local nucleotide, the trinucleotide, has a very profound impact of the mutation rate, and we are trying to consider the combinatory like impact of the epigenetic marks and the local nucleotide context to have a better fit of the background model.
I see. Thanks. And then before I guess other people ask question, you mentioned I guess ECLIP data and RNA binding protein, which I really don't know as much about. So I assume that it control a lot of protein translation. And it seems like you already analyzed a lot of how they may affect transcription, or maybe like RNA degradation is my guess, because they
Yeah.
seem to control RNA quantity. But have you guys also analyzed protein quantities? Are such data available?
Well, that's a very good question. Yeah, currently we are focusing more on the like RNA-seq, that's the transcriptome level quantifications. We haven't go to like the protein level, the true protein level quantification, like using the mass spectrum data. And so it's approximation. We don't have that huge data source for the protein level quantifications. So we have to confine our analysis mostly on the transcriptome analysis.
I see. Thanks. If anyone has any question, feel free to ask directly, or through chat, I can help you ask as well.
Yeah, for our annotation actually is purely online, and you can check the link from the ENCODE paper, and you can like feel free to download like the enhancers or the promoters and their different types of networks and their changing dynamics from different conditions.
Yeah, I think all these resources will be very helpful. I guess in closing, I guess one last question, Jing.
Yeah.
What's your favorite epigenetic mark, and maybe one that's really underlooked by a lot of us that don't know epigenomics that well.
My favorite H3K27AC, that is our active epigenetic mark in the genome that has been widely accepted as a indicator of enhancer regions. So if we are trying to find enhancers and we can only do one like ChIP-seq data, I will say probably like my current choice is 3K27AC. That's probably the best indicator of enhancers. But as we know more about the functions of the histone modifications, and we probably have an alternate choice, but currently that's my favorite.
I see.
I have a question too, going off that.
Yeah.
Is there any data, and I might have missed this, but like documenting methylation of transcription factor binding sites in different tissues? We talked a lot about histones, but like direct methylation.
Yes, there is. I think currently in ENCODE 3's companion papers, the transcription factor paper that is talking about the like integrating methylation data with the ChIP-seq data. And then they have some very interesting like discoveries so that the methylation seems to playing some role, some interaction with the transcription factors that are showing in distinct like states. So you're welcome to check into like this row of this paper. Like there is one paper that is mainly focusing on transcription factors, and they talk quite a lot about the methylation data there.
Cool, thanks.
Okay, I think if there's no other questions, let's thank Jing again for a wonderful talk and the foray into epigenetics. I think we all learned a lot, and really grateful for the talk, Jing. Thank you so much.
Thank you everyone. Have a great day there. Bye bye.